Well, good afternoon, and thank you for sticking around. By the way, I love the term biological uncertainty principle. I can't beat that. Hello, everybody. My name is Pujar. I'm the Chief Operating Officer of Tessera Therapeutics. Very happy to be presenting here about our research, and I thank the organizers for this opportunity. Our mission is, like many in the room here, is to cure disease by writing in the code of life. And that's gene writing is what we do. We have a platform that is designed to make virtually any change necessary to address disease. Another side of the platform is delivery, which is just as important as the gene writing platform. And we have non-viral delivery capabilities which enables differentiated in vivo genetic medicines. With these, with the gene writing and delivery, we've come up with differentiated programs that address a wide variety of unmet needs with blockbuster potential. We have a broad capability set in-house. We are vertically integrated, so that includes GMP manufacturing and digital capabilities in-house, and we have an extensive IP estate. So our platform for gene writing leverages a mechanism called TPRT, or target prime reverse transcription, which is a mechanism that evolved with retrotransposons. So this platform has two components, a gene writer protein and a template. The gene writer protein is comprised of multiple domains. These domains conduct TPRT over 4 steps of RNA binding, DNA nicking, and then reverse transcription. We sample domains across the tree of life and put these proteins together. We also create templates that provides the message to be reverse transcribed into the genome. And we screen these in high throughput systems and in vivo systems, and they're all dependent, of course, on the disease that we're tackling. So with these, we have come up with 3 kinds of gene writers. One is a rewriter, which allows us to substitute single nucleotides or small segments of DNA, including precise corrections of insertions and deletions. We can also write exons with single nucleotide precision at very specific target sites in the genome. We can also write whole genes that can enable restoration of gene function or deliver new instructions. On the other side of the platform is delivery. We have an all-RNA composition of matter, and this allows us to use non-viral delivery technology, which is lipid nanoparticles. These lipid nanoparticles are comprised of an ionizable lipid, which is kind of the business end of the LNP, and we have an extensive chemistry effort to screen and develop a library of ionizable lipids for specific applications. We also use targeting strategies for some of our tissues, and these comprise of a wide range of binders, cell surface antigen targets, and et cetera. And then, of course, this LNP is a 4 or 5 component system, so a very complex formulation problem. And so this 5-dimensional composition optimization is yet another capability that we've developed. With these delivery vehicles, we have achieved highly efficient delivery to the liver, to hematopoietic stem cells and T cells in non-human primates. For liver, we can get to more than 90% of hepatocytes. And if you compare that to what a target editing level might be for some of the diseases we're tackling, it's 10 to 30%. So we can deliver to cells well in excess of what the target is. For hematopoietic stem cells, we're very proud to have achieved 95% delivery to long-term hematopoietic stem cells. These are the rarest of the cells in the bone marrow, which are responsible for the entire milieu of cells in the blood. And there, the target editing levels are about 20%. And lastly, for T cells, we can get to about 40% of T cells, and the target level there is minuscule, 1% or even less sometimes. And so we haven't even bothered to optimize that further. So with these capabilities of gene writing and delivery, we've come up with programs that represents potentially best-in-class or perhaps even first-in-class treatments for diseases with large unmet medical needs. So the first one is alpha-1 antitrypsin deficiency, which is the largest liver monogenic disease. The second one is sickle cell disease. We're very excited about this. It's the most common lethal monogenic disease. Phenylketonuria, which is a common inborn error of amino acid metabolism. And of course, in vivo CAR T for both oncology and autoimmune. We have highly differentiated in vivo approach, and we now have non-human primate proof of concept for many of the key programs. And these programs allow for rapid clinical POC and accelerated development once in the clinic. So let me go over some data for each of the programs, starting with alpha-1. I'll get right to the non-human primate data here. And of course, these are surrogate reagents because non-human primates don't have alpha-1 antitrypsin deficiency. So with surrogate reagents, which in many cases are less effective than the human reagents, about 2 to 3-4 less effective, still here we can achieve a rewriting efficiency of 79% in hepatocytes at the SERPINA1A gene. And that's kind of validated with the amount of mRNA transcripts that we see in the blood. So 84% of mRNA transcripts have the corrected sequence. To our knowledge, this is one of the first programs to demonstrate this kind of correction in non-human primates in the SERPINA1 gene. We have developed a proprietary LNP to go along with this. We've done an extensive amount of screening and come up with an LNP that's highly efficient and has a very favorable tolerability profile. And we've tested this lipid in more than 100 NHPs at this point. The PK profile of the lipid is shown on the right side. And our half-life of the lipid is as good as any of the leading lipids in the field. We're now advancing the AATD program into the next stage of development. We've finished GMP manufacturing. We've also completed all of the IND-enabling farm tox studies, and we're on track for multiple CTAs and IND filings in the fourth quarter. Quickly going into PKU, very similar to the AATD program. We get very high levels of rewriting efficiency in non-human primates, again validated by mRNA transcripts. And here the threshold for correction is even lower than AATD. Only about 10% correction is what is required to restore phenotype, and we exceed that quite extensively. Going to sickle cell disease, here we're targeting the long-term hematopoietic stem cells in the bone marrow. But before going into the delivery part, here we show you in vitro correction of the sickle mutation to wild type, and we can restore almost 100% of these cells into the wild type mutation, which then results in near complete restoration of normal hemoglobin in patient cells. And these then, when tested under hypoxic conditions, show no sickling as what you'd want to see. In terms of delivery, we get to, as I mentioned at the beginning, 95% of these long-term hematopoietic stem cells, both in mice and in non-human primates. And when we combine the gene editing and the delivery, we can get up to about 50% editing in these long-term hematopoietic stem cells, which exceeds what is required to get a clinical benefit. modeling of this kind of correction suggests that this will result in 80% of red blood cells that are corrected, because the half-lives of the sickle RBCs is much smaller than the ones that are corrected. So net, you'd get about 80% corrected red blood cells in the blood. We haven't stopped here. We continue to make further improvements, and very excited about where this is going to take us. We're standing on the shoulders of giants, but the ex vivo therapy, as you know, is not reaching all the intended patient population. One key feature for the field, which I'm very pleased to share with you all, is that when you have non-human primate proof of concept, the probability that there is going to be human proof of concept is extremely high. This is a compilation of data for mRNA, siRNA, and genome engineering candidates, and you'll see that the probability of success is 70% or greater, and when you get to genome engineering, it's nearly 100%. And so we're very excited about where we are with these three programs, with NHP, POC, and we're excited to bring this all into the clinic. Before I close, I want to spend a few minutes on our T-cell efforts. And this morning, we were very pleased to learn that ARPA-H had included us in the list of fundees for the Embody program. So we're very excited about working with ARPA-H to bring this forward. Here, we're delivering our lipid nanoparticles with the gene writer to T cells in vivo. And we can do both the small edits to either knock out a gene. For example, in the left panel, we're knocking out beta-2M, and we get very high efficiencies, and we've gotten even better than this. But in addition, we can also insert a CAR gene into T cells using a template that's much bigger than your typical guide RNA, and we can get up to about 6% of T cells written with the CAR gene. And so we can multiplex now with both writing in a CAR and then knocking out several other genes. Using this technology, we have demonstrated that we can eliminate tumor burden in mice. So in this case, I won't go through the specifics of the data here, but you can see that we get an increase in CAR T cells, and that leads to a reduction in tumor burden, which of course you don't see in the control animals. And then similarly, we can take the same technology in a different model, demonstrate B-cell depletion, which is an application we need to see for autoimmune disease. With that, let me come to a close. We have broad capabilities in gene writing, writing in small or making small changes to the genome, or writing in full genes into the genome. We have extensive delivery capability to liver, hematopoietic stem cells, and T cells, and then growing from there on. This has resulted in highly differentiated programs with transformative potential for some of the largest genetic diseases. And all of this is done with a very high caliber team. Very proud to be part of that team and with strong execution. So with that, I thank you for your attention, and I'll be around tomorrow as well for any discussions.
Thank you to Tessera Therapeutics.